All right, let's get right into it. We're going to take a tour of one of the most amazing factories on the entire planet, and it's running inside your cells right now. Today, we're going to break down exactly how our bodies turn the food we eat into the actual energy that powers our every single move. I mean, really think about that for a second. How does that even happen? How does the chemical energy that's all locked up inside your lunch get converted into the fuel you need to think, to move, to live? Well, that's the big question we are answering today. So, to understand this energy factory, you first have to meet its most important employee. Or maybe it's better to think of it as the currency that pays for literally everything. Let's talk about ATP. What is just so incredible about ATP is how universal it is. I'm talking from a tiny little bacterium all the way up to a blue whale. This is the molecule everyone uses. It's like the dollar or the yen of the cellular world. Whenever a cell needs to get some work done, it spends ATP. Simple as that. So here's the whole point, the main goal of this entire process. Cellular respiration is how we take the energy stored in molecules from your food, glucose is the classic example, and use it to recharge a massive number of these little ATP batteries. That is the mission. Okay, so our factory tour starts with the very first step in this whole energy-releasing pathway. It's actually so fundamental, it happens before we even get inside the main powerhouse of the cell. Yep. Glycolysis. You can think of it as the universal starter kit for energy production. It all goes down in the cytosol. That's kind of the jelly-like stuff filling the cell. And it doesn't even need any oxygen. The whole process is about taking one molecule of glucose and just splitting it right in two. So what do we get from this first step? Well, we get a tiny bit of ATP. Think of it as charging just a couple of batteries. But way more importantly, we get 2 molecules of pyruvate, which is the fuel for the next stage, and these super important molecules called NADH. You can think of those as little taxis for high-energy electrons. And hey, before you start to get overwhelmed by all these new terms, just relax. The point here is not to memorize every single chemical reaction. You don't have to. The official guidelines even say so. We're just focusing on the big picture, what goes in, what comes out, and why it all matters. So, phew! All right, now that we've split our glucose, it's time for the main event. We're headed inside the cell's famous powerhouse, the mitochondrion. So those 2 pyruvate molecules we just made, they get escorted into the very heart of the mitochondrion, an area called the matrix. And in there, they get prepped to enter this really complex series of reactions. You might have heard of it. It's called the Krebs cycle. So the Krebs cycle spits out a tiny bit more ATP, and it also releases the carbon dioxide that you're literally breathing out right now. But you want to know its main job? Its real purpose is to strip away a ton of high-energy electrons and load them onto our electron carriers, NADH, and a new one called FADH2. It is all about harvesting that energy. Okay, so let's properly introduce these guys, because they are critical. NADH and FADH2, there are electron taxis. Their entire job, their whole purpose, is to pick up these energized electrons from the first 2 stages and shuttle them over to the final stage, which is where the real magic is about to happen. This is it. This is the climax of the whole story. All of that breaking down and shuffling molecules around has been leading up to this one incredibly efficient process. This is where we make the vast, vast majority of our ATP. So all our NADH and FADH2 taxis finally arrive at their destination, a series of proteins that are embedded in the inner membrane of the mitochondrion. This whole setup is called the electron transport chain. They drop off their high-energy passengers, and that single act sets the final, most powerful stage in motion. Now, as these electrons get passed down the chain, kind of like a hot potato, the energy they release is used to do something pretty amazing. It actively pumps protons out of the matrix and into the space between the 2 mitochondrial membranes. This builds up a massive electrochemical gradient. And you know the water building up behind a dam analogy? It is perfect. You're building up this immense potential energy. So, how do we cash in on all that stored potential? Well, first, we pump the protons out. Then, the only way for them to get back in is through this spectacular little molecular machine called ATP synthase. As the protons rush back through, they literally turn a part of this enzyme. And that physical, mechanical energy is used to slap a phosphate back onto ADP, creating, you guessed it, ATP. Lots of it. And this is just a beautiful example of how structure is linked to function in biology. Why is that inner membrane so wrinkly and folded up? Well, it's to dramatically increase the surface area. This allows for millions of these electron transport chains and ATP synthase factories to operate all at once. More factories means more energy. So what's the final count? What's the payoff here? Well, while the first two stages gave us a measly 2 or 4 ATP combined, this final stage, oxidative phosphorylation, it churns out somewhere around 30 to 34 ATP molecules for every single glucose. Now that is the massive payoff we were waiting for. Okay, so now that we've seen the main assembly line, let's just zoom out for a minute. What happens when things don't go exactly according to plan? And what are some of the other kind of surprising outcomes of this whole process? Well, think about this. Oxygen is the final stop for those electrons. If there's no oxygen around, our electron taxis have nowhere to drop off their passengers, and the entire assembly line just grinds to a halt. But cells have a backup plan. It's called fermentation. It doesn't make much ATP, but it's enough to let glycolysis keep running, providing just a little trickle of energy to survive until oxygen shows up again. Now check this out. Here is an absolutely incredible biological hack. This whole process can be intentionally made less efficient. By decoupling the flow of those protons from the ATP-making machine, the energy from that gradient gets released not as ATP, but directly as heat. And this is a super important way that mammals and birds are able to regulate their own body temperature. And that leaves us with one last thing to think about. The warmth that you can feel in your own body right now, it's not an accident. It is a direct and sometimes very deliberate byproduct of these billions of tiny warring energy factories inside every single one of your cells. Kind of makes you think, doesn't it?